Hi everyone, this week's lesson is about microscopy and gram staining. So we're going to be talking about compound microscopes, which are also known as light microscopes, and they use lenses to bend light rays to bring an image to your eyes. So there's two lenses that the light goes through to get to your eyes. Here the objective lens and here the ocular lens. And on that journey, the light rays from the object cross before they reach the eyes. So the image that you see will be upside down and reversed. So the parts of the microscope are the stage, which is the surface here where you put the slide. It has an aperture, which is a hole that lets the light reach your slide. And the next part to talk about is the condenser, which has a diaphragm or an iris, and that adjusts and concentrates the light that's coming from the light source here and up through the microscope into your specimen. And then the body tube of the microscope has the upper end and the lower end. So the upper end is the ocular or eyepiece lens, which magnifies 10 times. And the lower end has the nose piece, which is that rotating part at the bottom of the body tube that has the objective lenses on it. So the part that you rotate. And there's typically 4 objective lenses on a light microscope. The first is the scanning objective lens, which magnifies 4 times. The low power objective lens magnifies 10 times. The high power or high dry objective lens magnifies 40 times. And the oil immersion lens magnifies 100 times. So you have the 10 times from the ocular lens, and then you have to multiply that by whichever objective lens you're using to get the total magnification that you're magnifying your image. So for example, the magnification for a high power lens alone is 40 times, but the total magnification using a high power lens is 10 times 40. So 10 from the ocular lens times 40 from the high power lens. So your total magnification is 400 times. And then you have your focusing knobs, the coarse adjustment knob and the fine adjustment knob. So the coarse adjustment knob is the bigger knob, and it's for a bigger movement. So that either moves the body tube or the stage up and down, depending on what type of microscope you have. It moves either the body tube or the stage up and down to bring the lenses closer to your specimen. So that one you move it slowly, and you have to keep checking outside of the ocular lens, like look outside of the microscope to make sure you're not going to crack your slide when you move it up. And then the fine adjustment knob, when you get the coarse adjustment close enough, you can use the fine adjustment to kind of fine tune your focus. So I found this video that is a really good overview of the compound light microscope. And this is using the same exact type of microscope that we have in our labs at GCU.
This video is an introduction to the parts and operation of a compound light microscope. Once you transport your scope back to your station, remove the dust cover, fold it up, and place it in an out-of-the-way location to keep your workspace uncluttered. Whenever you transport your microscope, you want to make sure to hold it both by the neck and the base. This scope has a nice handle right in the neck, and then have your other hand beneath the base. Depending on the type of scope you have, you might need to plug a loose cord into both the scope and the outlet, or if you have a scope similar to this, the cord is attached to the microscope and you just need to unwind the cord and plug it into an outlet. Compound light microscopes typically have an on-off switch in the base. This switch lights up when I have it turned on. Understanding how to adjust the amount of light that's shining through your specimen is an extremely important skill to have. Too much light and your specimen can be washed out. too little light, and there's not enough illumination to see it. Adjusting the light allows you to manipulate contrast. That's the difference in the visibility of your specimen versus the background. Most compound light microscopes have a dial that will adjust the light level from the illuminator in the base. Another way that your scope allows you to adjust the amount of light shining up through the specimen is through manipulation of the iris diaphragm. This mechanism is located directly beneath the stage, and you twist it to open and close the iris. A mechanical stage is a very handy feature to have on your scope. You can place your slide into the slide holder and then adjust the position of your slide using the coaxial adjustment knobs, usually hanging off the side of the stage. This way you can center your specimen very easily. The focus knobs raise and lower your stage, and the vertical position of your specimen is what determines whether it's in focus or not. On this scope, the coarse focus can be adjusted by turning the part of the knob that's closest to the body of the scope. The fine focus can be adjusted by manipulating the outer portion of this knob. Doing so fine-tunes the image of my specimen. Compound microscopes have either a single ocular lens or a set of binocular lenses. The ocular magnifies the specimen 10 times actual size. The objective lenses are located on a rotating nose piece. Each lens hangs down from this nose piece and can be turned and clicked into position. The shortest objective lens is called the scanning power. It has a red band around it, and it magnifies the specimen 4 times actual size. However, since I'm always looking through both the ocular and the objective to view my specimen, I multiply the powers together to get my total magnification. So the ocular magnifies 10 times, the scanning magnifies 4 times, meaning at scanning power, my total magnification is 40 times actual size. The next objective lens is the low power lens. It has a yellow band around it, and it magnifies objects 10 times actual size. So the total magnification when using my low power lens is the ocular, which is 10 times, multiplied by the low power lens, which is 10 times magnification as well, for a total magnification of 100 times actual size. Now when I switch to the next objective, it looks kind of funny. The lens is covered up with a finger cot. In the lab where I teach, these scopes are primarily used for microbiology classes. And in micro, we're mainly using the oil immersion lens, the black and white banded lens, for viewing at high power. And the oil used with this lens can damage our high dry power lens. That's why we cover it up. But for now, we need to uncover the lens and take a look at it. I can dispose of that finger cot, and now I can see my high dry powered lens. This lens magnifies specimens 40 times actual size, but again, I'm always looking through the ocular and the objective at the same time, so my total magnification at high dry power is 400 x10. Compound microscopes typically have 3 or 4 objective lenses. If you do have a 4th lens, that would be the oil immersion lens. It has a black and white band around it, It magnifies objects 100 times actual size, so together with that ocular lens of 10 times magnification, I can get a total magnification with oil immersion of 1,000. An important warning: whenever you are using your high dry objective or your oil immersion objective, these are your two longest lenses, never, ever, 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 ever adjust the coarse focus. When you're using these longer lenses, you're only allowed to use the fine focus, again, that knob that's set within the coarse focus knob. Manipulating the coarse focus when using these longer objective lenses will at the very least take your specimen out of focus immediately, and worst case scenario could smash the lens into the slide, damaging the microscope. When you're all done, it's important to put away your scope properly so that it won't get damaged. You do this by making sure that the shortest objective lens is pointing down the scanning power. Then you dial the coarse focus down all the way to lower the stage as far as it can go. This way, we know that the lens is not going to smash into the stage. More seriously nerdy, amazing free stuff at scienceprofonline.com. Go there.
I want to give you some microscopy terms to know. So the first is parfocal, and a microscope that is parfocal is one which, if it is in focus with one objective, when you rotate that objective, it still remains mostly in focus. That would be parfocal. And then parcentric means that when the object is in the center of view, it will remain there in the center of view even when the objective is rotated. The third term is field of view. So when you look through the lens of a microscope, you'll see a circular area, and that diameter of that area is your field of view. And then finally, the depth of field is the range of depth that a specimen is in acceptable focus. So a microscope that has a thin depth of field, you would have to be continuously focusing up and down to be able to view a thicker specimen. So the field of view and the depth of field, those both decrease as you increase your magnification. So another quick video that I made in the lab is showing you how to use and focus a microscope. So always make sure that the 4x objective is in position when you're starting out. And you're going to want to turn that coarse adjustment knob to lower the stage all the way before you place the slide on. So you put the slide onto the stage with those stage clips and fit it right in there. And then you can adjust the oculars to fit your face. And those numbers there are a setting, they're markers, so you can remember the setting for future adjustments if you want to. So you use the stage controls to move the stage forward and backwards and left to right. And you should never rotate the objectives while the stage is all the way raised. You would lower the stage first and then change the objectives. So focus using the coarse adjustment knob first, and then fine tune it with the fine adjustment knob when you get close enough. In microbiology, we often use oil immersion to look at our very small bacterial samples. So with oil immersion, you use the 100x objective lens and you use oil. So first you focus the slide with the 40x objective with no oil, and that lens should be almost touching the slide as you get it focused. And then you turn the nose piece partly so that it's between the 40x and 100x lenses. And then you would place one drop of oil onto the center of your slide and then rotate the nose piece all the way so that the 100x tip is in the oil. So once you're there in that position, you don't touch the coarse adjustment knob because you could damage the microscope by adjusting the stage too far up. So at that point, you would only use the fine adjustment knob if necessary to fine tune your focus. And this picture is showing you the 100x objective with and without oil. So if you use it without oil, a lot of light from the microscope is lost out the sides. But when you use it with oil, most of the light then enters the lens. So that's what makes the oil a more effective way to view very, very small specimen with the 100x lens. Now we're going to talk about gram staining. So several stains have been developed to provide contrast and to make it easier to see bacterial cells under the microscope. So simple stains use only one dye, such as methylene blue, and that allows the observer to see the cell size, the arrangement of the cells, and the shape of the cells under the microscope. Differential stains, on the other hand, use two or more dyes, and that allows us to distinguish between different types of bacterial cells or cell structures. As you can see in this picture, the two dyes of the differential stains can help us tell apart two different strains here. The most common differential stain is the gram stain. And the gram stain distinguishes cells based on their cell wall composition. So there's gram positive and gram negative cells. Gram positive cells, as you can see represented in this picture, have a cell wall composition with a lot of peptidoglycan. So the cell walls have a thick layer of peptidoglycan mesh on top of the plasma membrane. Gram-negative cells, on the other hand, have very little peptidoglycan, and they have lots of lipids with an outer membrane. So you can see in this picture representing the cell wall of gram-negative cells, very little of that peptidoglycan mesh, and they have the outer membrane on top of it. So the technique for gram staining is a pretty simple process. The first step is taking your bacterial smear and staining it with crystal violet, which is a basic dye that stains everything purple. The second step is adding the grams iodine mordant, which is added to help set the dye. The third step is that the smear is then rinsed with a decolorizer, such as 95% alcohol. And this step, what happens is it washes away the diiodine complex from the gram-negative cells. And that happens because the alcohol damages their cell membrane. But it doesn't wash away the dye from the gram-positive cells because they have that thick layer of peptidoglycan. So after the alcohol wash, you end up with the gram-positive cells keeping their stain purple, and the gram-negative cells here have been decolorized. So the fourth step is safranin is used to counterstain the gram-negative bacteria. So all the gram-negative bacteria, their color has been washed off. So this is a counterstain, and it stains them pink. So the end result that you end up with is that all your gram-positive bacteria are stained purple, and your gram-negative bacteria are stained pink. And that thick layer of peptidoglycan in the bacterial cell wall is what makes the gram-positive bacteria retain that purple dye. So to help you remember, gram-positive, purple, plenty of peptidoglycan. And why should we care about whether bacteria are gram-positive or gram-negative. Why do we need to determine that? Well, it's actually very important in medical labs. Gram stain helps the clinical lab technician to identify the organism in an infection. And the gram stain results can be important for treatment decisions in a patient as well. So gram-positive and gram-negative bacteria often differ in their susceptibility to different antibiotics. So it would help the doctor to know what antibiotics to prescribe if they know the infection is a gram-positive or gram-negative bacteria. So for example, gram-positive cells are more susceptible to penicillin and gram-negative cells more susceptible to tetracycline.